Okay, well now we're going to have Dr. Kyle Van Houtan, who leads the Marine Turtle Assessment Program. So this is a great opportunity. His goal is to generate science to understand extinction and to encourage ethics to prevent it. So that's a great goal, in my opinion. And he's focused on marine population ecology, conservation. He's been featured in a whole list of magazines that are, and books, or I should say periodicals, but Nature and National Geographic are just kind of a couple of the highlights. So you can just imagine what the rest are like too. So without any further ado, I'd like to introduce Kyle Van Houtan.
Hey everybody, how's it? Last night we spoke in Kihei, and Ka'au is the head educational guy at the whale sanctuary there, and he gave this eloquent Hawaiian chant, and I said, I'm just going to say, how's it? I came to Hawaii in '96. My mom actually went to UH Manoa, of all places. When my father was in Vietnam, she came here to be sort of as close as she could to me. So they lived on Date Street in Mo'iliili, if anyone knows where that is. I came here in '96. I actually lived in Makawao, upcountry. So my Hawaii experience for the first couple of years was Paniolo country. It was upcountry. It was lush. It was green. And I have to confess, I'm a bird nerd. So I worked on birds for the longest time. It's only been recently that I worked on sea turtles. I'm a population ecologist, and I came to Hawaii because it is the epicenter of extinction, with all sorts of unique plants, animals, fish, coral that don't exist anywhere else in the world. They're endemic here. They're here and nowhere else. And it's been a kind of bad rap the past 100 years or so for native species here in Hawaii. I came here to work on the native honey creepers. So I worked on Mauna Kea, on the palila bird. That was the first bird, the first species for which the Endangered Species Act protected land as critical habitat. Landmark case, the palila bird here in Hawaii. You can be proud that it was a Hawaiian bird. That was the first example where the Endangered Species Act preserved land and said, the Army, you cannot use this to blow things up because there is a bird here that's nowhere else in the world. And if you do that, it's going to go extinct. Probably the sad thing is it probably is going to go extinct anyways, but at least we can say it's not because of the Army, which I think is something that's a good thing. I moved here to Maui to work up in Haleakala, so I spent all my time, I lived in Makawao, but actually I lived up in Hanavi, if anyone knows where that is. It's about 8,000 feet up. It's very cold, it's very wet. Sometimes we'd go up there, they'd drop us off in a helicopter, and it would rain for 3 weeks. And I had these things, which I don't even know if you know what they are, Gore-Tex socks. I don't even wear socks now, let alone Gore-Tex socks. But I pulled them out of storage and I was like, what is this crazy thing? They were so uncomfortable and they were not waterproof. So we would walk around and just get soaked to the bone. We'd wake up and there'd be frost in the ground. It was like, this is Hawaii, what? But it was gorgeous. The trees, the ohilehua up there, the fern forest, they fenced it off, there's no pigs. And the kohe, the Maui parrotbill, the iiwi, the apapane, the akepa, there were nukupu, I think, up there when we first got there. It was a gorgeous place, and it still is. If you've never been there, it's a brutal hike, but maybe I'd recommend it. You have to get special permission. But now I work on sea turtles, and I live on Oahu, so I'm back over here. It's great to be back on Maui. We've been in the water today and yesterday, and seeing a lot of really healthy looking turtles. So it's happy to see that. I'm gonna, I think, can use this dude. Awesome. I'm gonna talk tonight about, I was supposed to do that by the way. I'm gonna talk tonight about fibropapilloma tumors, but I'm gonna give you kind of a little popcorn of some different research projects I'm working on to show you the kind of breadth of things we do to generate science, which is interesting for scientists and ecologists, but also helpful for management and for conservation. So these right here, think about what those might be. These are actually data. And when I came here to Hawaii to work for NOAA to do turtles 3 years ago, I kept on seeing this phrase, the Hawaiian green turtle was driven to near extinction because of the tourism restaurant industry. And I was like, wow, that's amazing. Because my parents lived here then, and they never saw it in the restaurant. So I went, actually, I was sort of believed it, and I sort of didn't believe it. And I'm a scientist, so I said, well, I want to see the data. So the data in this case are restaurant menus. So I went back and found restaurant menus. We found 400 or something between 1920 and 1974. They had a legal fishery for green turtles here from 1946 to 1974, and they actually kept records for that. And we've been analyzing it, but one of the things we don't have in our NOAA database is what they were selling in the restaurant. So I wanted just to go back and find out if this was true. Were they selling turtle in the restaurants? And so we first put together, here you can see time on the bottom, and here's the frequency. So we figured everybody would understand what beef is, right? And it's kind of a baseline. So what might you think happened in early 1940 where the frequency of beef was maybe rationed and went into decline? World War II, right? So that seemed to be a pattern that made sense to us. Beef's pretty popular. One would be it's on every menu that we found in that time period. And then we said, ooh, let's look at like the big fish, you know, like billfish and tuna. Back in the old days, before Hawaii was a state, it wasn't really on any of the menus. Anyone think of why? We just didn't have the big boats that went out to deep sea, right? It probably is, I mean, the menus reflect a lot of things. They reflect the industry, they reflect taste, what people wanted to eat. But look what happens when you look at reef fish. They used to be on every menu, and some menus would have 2, 3, 4 species of reef fish. I learned species that were edible that I didn't even know we had in the water here. Kumu, veke, ulua everyone knows about. They have them at the Mountain Ocean Center, but the goatfish that has little whiskers, kind of looks like a catfish. It's a fish I'm much more familiar with eating. People, when you talk to a local person or someone who has Hawaiian ancestry, and you say kumu or veke, it's W-E-K-E. And I was like, what's weak? I've never seen that before. And they go, oh, kumu, oh, veke, it's so, oh no. You know, they love that, but it's just not really around anymore. So when we, it's just really amazing. These menus are just showing us what we knew, that these species declined. By the time Hawaii became a state, the reef fish were not commercially viable. That's amazing. So turtles, we got into this for turtles. Look at a turtle. It was on a few menus, but never very many. So what we found in talking to people that actually fished in the fishery, the people that worked in the markets, was that yes, there was a fishery, yes, it was commercial, but it was small scale, and they sold to local meat markets like Star Market or Wing Sing Seafood Market, a lot of it based in Chinatown, Haleiwa Farmers Market, and it was a local clientele. Some restaurants, yeah, you know a place you could go to get turtle soup, or you know a place you could go to get turtle steak, but it was not the tourism industry. And this is important because as we talk about delisting, we have to establish that we have mitigated historical threats. And this seems to be that the main historical threat was a local, small-scale fishery for local consumption, not hotels. So, hawksbills in the northwestern Hawaiian Islands. Northwestern Hawaiian Islands, they're not supposed to be there, what? So one of the things we do is, this is a paper that's coming out in a few months. We went back into the historical literature, like Navy records, naturalists from the 1700s, from the 1800s. Somebody that worked for me had to dust off her French and read some French naturalist records from La Perouse and from some other voyages. But here's the Northwestern Islands. You can see here, we're down here, this is what we call the monument. And you can see hawksbills were there historically. Obviously, we don't have pictures of them back then. And in some cases, these people weren't trained to differentiate between what's a hawksbill and what's a green turtle. But we had this really interesting record where a ship went out from Honolulu to Lysianski and the French frigate Shoals and brought back a lot of tortoiseshell. Tortoiseshell is a proxy for hawksbills because they did not sell tortoiseshell from any other sea turtle but hawksbills. So they brought back a lot of tortoiseshell. We've done some research on what a lot of tortoiseshell might mean, and we found that the plates, which we call scoots, that form the back of the carapace, if you stacked up a cubic meter of them, okay, just stack them up, that's almost 600 turtles. And that's what they did. They would rip the plates off through heating them or boiling them. So you can fit 600 hawksbill sea turtle scoot plates in a square meter. So what is a lot of tortoiseshell? Well, there was apparently quite a bit up there. Today we're not doing, it's a big place, the population is small, we're not looking for them up there, but we have found some pictures of them recently. This was a stranded turtle. This came through the tsunami. This was just cruising around the reef last year. And so we're trying to use creative means to say, how can we find out where this population is? This is important because this species is arguably the smallest distinct population of sea turtles on the planet. And we don't know very much about them. This paper doubles their known range. That's a good thing. It means they're out there. We just need to learn more about them. So, why do turtles bask? This is a data set that we got from a volunteer group that's been on Oahu up at Laniakea Beach. It's called Malamana Honu. And this dark blue line right here is a series of basking turtles through the months for the past 6 years. And you can see a kind of cyclical pattern here going. There's a lot of them, there's a few of them. This right here, these units on the y-axis, I just kind of transformed the numbers so I could plot them on the same plot. But you know, a lot, a little, so just kind of get them sort of this way. Now this light blue line, sort of like Carolina blue, is the sea surface temperature. Now what I've done is I've actually flipped it, so the peak is actually peak cold. So I'm calling it sea surface temperature stress, or cold stress. So what does this tell you? Well, we expect that ocean temperature kind of follows an annual cycle, right? But so does the basking. So when the water is the coldest, the basking is the most. When the water gets warm, they stop basking. When the water gets cold, they start basking. When the water gets warm, they stop basking. And you know, I can do that a few more years, right? So what does that tell you? They have basically a solar panel on their back. And they're reptiles, so they take on the environmental temperature. They get out to get warm. Now they can get a lot of benefits. We know that they can increase their body temperature about 5 degrees Fahrenheit just by hauling out for a few hours. But that can help them digest, it can help their immune system work a bit better, and they appear to be doing this in a really interesting way. Now I wrote some models here, which are basically just sine curves, if anyone remembers that from trigonometry. And this dark blue line is basically a model of that. And look how these are just kind of going right in concert with one another, just following it perfectly. Now these orange lines, there's 5 of them there, there's an overlap of 2 there. These are lines where we've established the timeline of lines in their bones, okay? You think about dendrochronology, which is the study of tree rings, and you're in the northeast United States where you have deciduous trees, they leaves change colors in the fall and they drop. Those trees, if you cut down a maple tree in Massachusetts and you counted, went from the outside along a line into the center of that tree, and if you had 15 rings, the tree is how many years old? 15 years old. It's 15 winters because it dropped its leaves, it wasn't photosynthesizing, it wasn't gaining energy, it stopped its growth, and it just kind of hunkered down for winter. These turtles are putting lines in their bones right when it's the coldest and right when they're basking, and that always seems to be right there. So there's something going on with these turtles that they're having this arrested growth, kind of like a tree does in the winter, and they're basking. They're telling us something about the climate and how it influences them. So this is from a couple different individuals, and if you put them all together, those lines in the bones sort of peak in April, May. This is just right when it's the coldest, right when they're basking. So kind of cool. We're putting this together and finding out ways how climate influences turtles on an individual level and at a behavioral level and their physiology, so it's kind of fun. So some more historical stuff, thinking about green turtles now. That's a big green bar for green turtles on top. The black triangles are where we have nesting today, and the size of them is proportional to the magnitude of the number of females that nest there. The circles are where they nested historically. So think about what we know. The numbers have gone up, and that's awesome. But they're really only nesting here in significant numbers. Also in Lake Santa Pearl and Hermes. But look at where they nested historically in big numbers. In a lot of places, and in the main islands. These little squares are archaeological sites where they found turtle bones. And you may say, oh, I know, Lahaina, there was a turtle that nested there. The gentleman over here was telling me he has some video of it. But there's very isolated spots, all of the main islands are starting to pop up, but not the numbers that were there historically. So it's something to consider as we talk about delisting, as we talk about recovery. We need to take a longer historical perspective to see what was normal, right? Okay, so now actually we're going to get to what I'm supposed to be talking about tonight. And that is fibropapillomatosis, or these tumors. Now this is a paper we actually published in the late 2010, not 2 years ago. You can go online and find it. It's called Land Use Macroalgae and Tumor-Forming Disease in Marine Turtles. And I'd encourage you to do that. It's free, and you don't have to pay for anything like that. So this is what it looks like. Has everyone seen this? Right? It looks like cauliflower kind of growing up. Pretty gnarly, I'd say, pretty nasty looking. It can get really bad, and I'll show you some pretty gross photos. This is on the big eye. I'm sorry, this is on Oahu. So it's a herpes virus, pretty well established by now. Herpes viruses, a lot of different kinds. They're implicated in a whole host of diseases. When I started doing this work, I started to do some trying to understand a bit more about herpes biology. Herpes is ubiquitous. Everybody has herpes in their body. If you've had chickenpox, if you had cold sore, if you've had mono, you know, the kissing disease should be called the herpes disease. If multiple sclerosis is even believed to be linked to it, because herpes comes from the Latin word, which is also what we get herpetology from. Does anyone know what herpetology is? The study of snakes and reptiles. Everybody says snakes though, but that's great because herpes, like H-E-R-P-E is Greek for to lurk, it's kind of nefarious. It's like to hide, to sort of wait for your prey to come by, and that's what herpes does. It lurks. You get a cold sore, it pops out, it's kind of gross, and then it goes away. And hopefully, if you're lucky, it'll come back months later. What it does is just waiting for the time is right, waiting for it's a good conditions for it to manifest itself. And that's what herpes kind of does. This is kind of a strange example of herpes because it's kind of the unherpes. The tumors come and they manifest themselves and they get bigger, bigger, bigger. They pop up in their flipper, they pop up in their eye, they pop up in their mouth, they pop up in their shell sometimes, and they don't go away. It's the unherpes. They manifest themselves and they stick around. Now, there have been some examples of tumor regression, but it's been very isolated. We only know a few examples of it. So this is a kind of family tree of herpes, if you've ever wanted to see that before. If you're up in your, on your Latin, you can see there's a whole host of species. Asian elephant is not Latin, and neither is human. But, you know, parrots get herpes, humans get herpes, monkeys get herpes, pigs get herpes, turkeys get herpes. What's important from this, the reason they did this is they studied the evolution. They said, well, this colonnid herpes virus, which is one for sea turtles, differentiated from the rest of them about 300 million years ago. And here in Hawaii, we have Colonia minus herpes, or I'm sorry, Colonia minus Hawaii. This is the virus here that specifically affects Hawaiian green turtles and causes the tumors we see here. And that diverged from the other herpes that just affects sea turtles 3 million years ago. So these viruses have literally been around for millions of years. But how long has the disease been around? A couple decades. So what does that tell you? The virus has been around in turtles and in Hawaii for 3 million years. But the disease worldwide really showed up. It had panzootic, which is a way to say pandemic for animals, panzootic levels in the 1980s. So why would that happen? Why would you have the cause of the disease around for so long, but actually the disease not there? Think about that. What's that? The time is right. Well, yeah, the authors of that paper, which I wasn't a part of, said that something in the environment is happening to promote the disease to make it make the timeline. So the diseases are complicated, and we're going to really be focusing on, today I'm going to talk about exposure. And one of the things that we have to consider is how do the turtles get the herpes? Just like humans get the herpes, from contact. And this is one way they get it. These cleaner fish, they haven't ever been to a cleaning station. You see 10 turtles just kind of hanging out, and a bunch of things are kind of picking food off of them and stuff. And this is an example of some fish cleaning off the shell, eating some algae off the back. There's been marine leeches, ectoparasites, things that kind of hop from turtle to turtle have herpes DNA in them. We've seen pictures of like wrasses. People have told me, and I've seen pictures of wrasses I forget which one it was. Rats is eating, browsing algae off of a tumor, you know, so they're carrying genetic material of herpes from turtle to turtle. Turtles also get a little frisky with one another, bump into each other, they can practice. Young adolescent boy turtles will practice, like one was a girl, and they'll mount each other and they do all that kind of stuff. When you've probably seen them foraging on limu, they're in the shallows, they're bumping into each other too, so there's all sorts of opportunities for them to transfer into contact with one another. This is what it looks like. This is over from Maui, and from South Maui over Kihei, and what we did is we looked at, the study that I'm going to talk about tonight is strandings. So these are dead turtles that are about to die, that wash up on the beach. We have about 4,000 records over a 28-year period, and this is a classic example. Most times that turtles get this disease, they get tumors in the eyes, about 90%, and it is fatal. It kills turtles by mechanically interfering with them closing their epiglottis, so they often die from pneumonia, or they're overloaded with salt because they can't extrude the salt from their eyes because they're blocked with tumors. They can be malignant, but oftentimes they are not. So these are just the raw, interpreting the raw data. These numbers are kind of small in here, but these are just 5-year periods. This is the demographics of the population. Here on this axis right here, we're going from small to big, which is kind of all other things being equal is young to adult. And on here is just the size distribution of the strandings over time, and then here's the disease rate. And what's important to see is that it's just not a uniform distribution. The turtles that are dying aren't equally of different age brackets, right? And importantly, we have this pattern here where when turtles first show up, if you remember Irene was saying they hatch predominantly in the northwestern Hawaiian Islands, they spend some time, a few years, out in the open ocean, and then they recruit to the main Hawaiian Islands, right? When they show up, do they have this disease? No. They're disease-free. The shells are clean, they're very pretty, kind of like they would look over the aquarium. And then, the longer they stay in the main Hawaiian Islands, they get sicker and sicker and sicker until they're subadults. And then what happens? They become adults and they start breeding. And then what do they do when they start breeding? They start going to the northwestern Hawaiian Islands. That way, sorry. And that is a place that's not inhabited. It's not influenced by humans, at least the way the main Hawaiian Islands are. So we have some interesting things going on here, but because we have this population which is non-uniform, we don't have an equal number of kids to parents, if you want to think about it that way, and the disease afflicts this kind of population segment, we have to do some statistics with it. Now fortunately, before I came here, I taught at Emory University, which is in Atlanta, Georgia, and right next to my building was the Center for Disease Control. And I used to teach with some of the statisticians, or public health statisticians, or biostatisticians. And this is exactly the kind of formulas that we would use to describe maps of influenza or sickle cell anemia, because most diseases have demographic risk factors. Like I always say, if we were to put a map of cataracts in the United States, if you didn't think of the demographic risk factors, you would say, holy crap, what's going on in South Florida, right? But the reason there's a lot of cataracts in South Florida is because of the population demographics, right? Babies don't get cataracts, right? And the population is the elderly population in South Florida, and that is the segment. If you do the same thing with a whole host of diseases, like I mentioned sickle cell anemia, it afflicts African Americans more than it does Caucasians. And that was actually an evolutionary adaptation to malaria, which is kind of fascinating, but not affecting us in the United States. So we have to do these demographic corrections. And it's important because when we apply this to like a time series, we want to describe the variability of disease over time. Is it going up? Is it going down? Is it bad here? Is it bad there? If you want to make a map, we have to do these corrections. So we kind of have to do our homework. The first thing I'm going to show you is a time series from 1980 to 2009 for all the Hawaiian Islands. And this may be familiar to you. You might say, oh, the disease was really bad in like 96, 95, and then it seems to be getting better. The incidence of the disease is going down. Well, let's break that down by island. Look at Oahu. And so we're taking, when I say all islands, that's actually Oahu, Maui, and the Big Island, because that's where we have the stranding programs that are really in effect. We have some data from Hawaii, we have a few from Molokai and Lanai, but really the bulk of our data, so we're going to stick to those, the main 3 inhabited, at least inhabited by human islands. So that Oahu trend looks a lot like the all-island trend, and that's because most of our data is from Oahu, so that's just kind of an artifact of that. But what happens if we say, oh, you know, Oahu is kind of a diverse place. There's Kaneohe Bay, there's the leeward side, there's South Shore, there's Mauna Loa Bay, there's the North Shore, there's Kahuku Coast, there's Waimanalo. If we break this down as a hydrologist might break down the island into hydrographic regions, look at the trends that pop out. There's no one trend for Oahu. There are all these different trends. It's going up, it's going down, it peaked. Like, this is, I live in Mauna Loa Bay, and it's kind of seems to have plateaued a little bit. South Shore, which is this whole area from Barber's Point to Diamond Head, it's kind of going up. The Waianae Coast, this is a really scary one. It's kind of spiking, and that's gone up since. North Shore, it's gone up, and it's kind of gone down. And so you can see which level do we look at? Which one is the right answer? Is it the yellow box? Is it the pink box? Is it the white boxes? Well, here's Maui, and there's the Big Island. I don't have a map of Kona and Hilo because basically you just draw a line down the middle of the Big Island and left side, right side, so it's pretty simple. There's the Hamakua Coast, but it's inaccessible for us. So what you see here is a whole host, and we ran some models, and what the models say is the white boxes are the right answer. So there's so much variability going on in the local sort of hydrographic region. That's what we must pay attention to, and that's important because what it says is there's no one trend, and this disease probably has a local influence of some sort that we need to pay attention to. So, one of the things that's really interesting about Hawaiian green turtles, if you've been here a while, they have a fidelity to local reef patches, right? They will sit in algal beds, and you may see the same turtle there, if you're lucky, for 20 years. I mean, it's not always going to be right there if you go in the water. Maybe some turtles are sort of dependable, but George Balaz, who runs a different turtle program, has these 5 different sites around the islands, and he's captured some of the same turtles for over 20 years. They have a pattern, and they have fidelity to these algal beds or different reef areas, and they're near the shore. Okay, they're near the shore. So the first thing I'm going to show you is a map of the disease. And we had this time series I did before, but that said, we need to go to these local hydrographic regions. I'm going to break it down. I'm going to throw away the differences in time, and I'm going to focus now on the differences in space, because I don't have enough data to do a time series for this little watershed. And that's what this is. This is a watershed-based analysis. Now when I'm doing this, these are the disease rates. So red is really high, and green is pretty much disease-free. The gray areas we didn't have any data for. And these are kind of like the a'upa'a, or basically like a watershed approach. Now it's important to say here that just because this whole area is orange does not mean that turtles stranded up here on the saddle. Or doesn't mean that we had way up Mauka some turtle stranding. What this means, I'm saying, is that perhaps there's an influence to that watershed from Mauka to Makai, right? And the Makai is where the turtles are, right? The turtles are stranding on the shore. And I'm just telling you this watershed, I'm assigning that, I'm making that watershed, that maybe there's some responsibility to bear upon that. So this is a map here, and you can see any patterns here. Congratulations, Kihei is the worst place. This means 93% of the turtles that have died in Kihei have this disease. 93% of turtles have tumors. That's really scary. The Kona Coast has basically been disease-free as a contrast. But Maui in general doesn't look so hot. It's a lot of orange and red. We're not really surprised about Pearl Harbor not being a very clean place. We're not surprised about Mauna Loa Bay. It's had a lot of problems. Kaneohe Bay used to have a sewage outfall pipe right in the middle of it, and they changed that, but still some problems. There's some things going on up here that we know about that are problems, but the leeward side, as you saw that one I showed you, the Waianae Coast, it was really low and it just spiked at the end. It still looks pretty good. Over this 30-year period, this is where Waianae is right here. But Kihei is ground zero. This is a turtle from Kihei, horribly, grotesquely afflicted with this disease. We see this a lot. If anyone's worked on a volunteer program here in Maui, the turtles that come on Kihei are really afflicted. They're not just having tumors like, oh, there's a flipper that has a tumor on it. No, we get this really disgusting A different turtle, this was from 3 years ago. A different turtle that just came out last year had a 5-pound tumor on its left eye. You know, so Kihei is a really bad place. Not only do most of the turtles have this disease, but they have it really bad. So this is the map. And what's important about this is that the neighboring areas have a similarity. And that's what this thing on the right said. This is talking about spatial autocorrelation. And this just means that there's clusters. There's clusters of areas that are sort of low disease and clusters of watersheds that have high disease. And that means if we're going to analyze this, we don't need to just say if a turtle strand's here, it's influenced by this, but it's also influenced by what's around it, right? There's a sort of spatial relation. Do you have a question?
I was wondering with West Maui though, because I was wondering about that because we have green and red.
Have green and red,
Right
yeah.
there, like how can that be? Doesn't that go against what you were
Those
just
are the data.
Yeah.
So what we basically have is the turtles. What we did is, you know, some of this could just be the influence of local currents.
Yeah, well that's what I was wondering if it was, yeah.
If a turtle strands right here on the border of this watershed, I'm going to, you know, assign it to one or the other, and this could just be an artifact of that. But what you have is you have some bad, okay, bad, okay. And if we did this maybe and broke it down, and maybe we had enough data to break it down, it would sort of vary. It would sort of, maybe it sort of flip-flop. But we have, you know, it's not good really on Maui, really ever. A couple places that looks okay. We can talk about that in a second.
Yeah, I guess I just wondered sort of like what he was saying is a terrain base. Because in
Could
that
be.
area of Maui, you've got some big valleys and cuts.
Some local effects, right? And this is a big map. A watershed can be a very diverse place. If you see, actually, you know, here on the north shore of Oahu, look how skinny the mouth of that watershed is right there. So, you know, there's some things coming together right here. But yeah, I've always looked at that and said it's interesting, you know, but Honolulu, it's kind of green up there as you get towards up to the top, and I guess this right here, I think we're like right here. We're right on the border. I should probably use that so my finger's not in the way. But we're right about there, I think. So as you go up, there seems to be a little gap, but it could just be a local current or something dragging the dead turtles. But we don't have a big problem with turtles stranding and dying, and then the currents taking them a ways before they die. That happens a lot on the East Coast because they have this big continental shelf. The turtles here kind of tend to die where they, or show up where they die. So a couple things that I've talked about already is telling us that the local ecosystem is probably influential for this disease. That herpes family tree I showed you, the virus has been around for millions of years, but the disease only a few decades. That's kind of peculiar. The behavior of these turtles, they have this fidelity to local patches of algae and reef areas. And then the strandings data shows us these clusters of disease areas and the clusters of disease-free areas. So I wanted to sort of take a unique approach and build a footprint for a land use footprint for each of these watersheds. And I got this cue from the Wildlife Conservation Society, who put these footprint statistics together to develop a map of the last wild places on Earth. And they did this using a kind of bunch of different data that was available to them, like infrastructure projects, like electricity lines, telephone lines, roads, population centers, population density, land cover. We track over time when forests are cut down. When forests cut down, it's probably an indicator that there is not a wild place. Night lights, have you ever seen a map of the Earth at night and you see New York City, the whole eastern seaboard is like bright white, and then like Nevada and central Las Vegas is really black. You know, that kind of gives us an indication of human population. They composite all of these data together and they give a map and they say, the Sahara is a wild place. Well, we already kind of knew that, you know, but it gets some interesting results. Now, what I did is I didn't do a map of the last wild places in Hawaii. What I did is what I call a nitrogen footprint, because I want to know the influence of nutrients coming from the land that's going out to the ocean from these watersheds. And I did is I composited a bunch of data from the state and data that we had to generate ourselves, but we wanted to sort of capture eutrophication. Okay, Hawaii has oligotrophic waters. That means there are few nutrients in them. And anytime you have an oligotrophic system and you elevate the nitrogen or the phosphorus in it, it dramatically changes that. If it's a lake in Arkansas, if it's a river in Niger, it changes when you add nutrients to it. And Hawaii is no different. It obeys the basic laws of nature like every other place. So we came up with this statistic, this footprint analysis of factors that generate, deliver, and retain nutrients in the nearshore ecosystem. Some of these are natural. Most of them are anthropogenic. So I'll just run through a few of these. Sewage injection wells. Has anyone ever heard of a sewage injection well here in Maui? It's a kind of a popular topic. Cattle grazing, cow pies, full of nitrogen. Urban cover, sugar and pineapple, no secret there. A little bit of sugar here in Maui still, as I saw, just about as much as here when I used to live here. Animal factories, we actually do have enclosed areas where people raise chickens and pigs, generates a lot of poop, a lot of manure, that goes into the ocean. Aquaculture and fish ponds, estuaries, wetlands, water courses. This is how these nutrients can be communicated to the reefs. Most of the water that comes from land to the nearshore area comes to groundwater, but some of it comes obviously through canals and perennial water courses and rivers and streams. And then fringing reefs and boat harvest kind of can hold the nutrients in, prevent flushing, and boats and the harvest can sort of churn things up and make it more available. So when we did that, here's the map of the disease that I showed you on the left. And I'm going to show you now the nitrogen footprint map, which is going to be colored the same way. And those maps look a lot alike. Okay, the map on the right, just look at the big island to start with. You can kind of blink your eyes and look at one with the other, and they look almost exactly the same. You have, this is Hilo right here, and we've had a lot of turtles. I think about 8 out of 10 turtles, maybe a little bit less than that over the past 30 years, have stranded with this disease. It's also very high in generating nutrients and all the activities in this statistic I put together. And then if you look on the Kona side of the Big Island, look, it's not a whole lot of disease and not a whole lot of nitrogen generating activities. That's changing, it's getting a little bit worse. Yeah, you have a question over there.
Just wondering if the nitrogen levels are recorded on land or in the water, like maybe like this Big Island, the west side. I was just trying to account for the difference there. Like, did you measure it in the ocean current or in the?
So what we did is we generated this land use from a bunch of land use statistics that we were brought together. And most of the data is from the state, some of it we had to generate ourselves. But nobody is recording nitrogen in the water in Hawaii. They're not doing it in any one place throughout time, and they're not doing it all over the place at one point in time. That's why we had to generate this, because those data weren't available. So we had to come up with something that would be a good substitute or a good proxy for that. And this footprint statistic was the best thing that we thought we could come up with. We've revised it a little bit since to make up for some areas of data we didn't know about, but this was the best thing we could come up with at the time. So there's a lot of sugar here in central Maui, certainly contributes to the orangeness here. But you can see that there's a lot of sewage injection wells here. Here there's some hog farms. There's a lot of sugar here, and there's still pineapple here. There's a lot of people living around here. There's a lot of sewage discharge from cesspools and septic tanks there. There's a lot of septic tanks here. Some agriculture up here. There's a wastewater treatment facility right here. Wastewater treatment facility right here. Wastewater treatment facility right here. Wastewater treatment facility right there. Kind of sounds like a broken record. But these maps have a lot of similarity. And what this says right here, it says that 85% of the differences in this map can be explained by that map. So what it looks like right now is 85% of this disease is responsible by land use. That's what the statistics say. Now we have to we can't just go by this correlation, we have to explain why that might be the case. And but before we get there, I just want to break down, so this is a sort of quadrant breakdown of nitrogen footprint on this axis and the disease rate on this axis. And so 0, none, 1, a lot, these are sort of scaled the same way. You can see these gray circles are Oahu, you got this big spread of them right here, which is kind of reflecting on these maps looking alike. The big islands, the orange triangles, you see there's this big cluster down here, low disease, low nitrogen. Then you had that one outlier up there, right? The Big Island map was mostly green and yellow, and that one big red spot right there. That's what that is. And then you have Maui, which kind of has really, these are all kind of high. So you have this just really, of the 3 islands, Maui's the worst in terms of the disease. That sort of says. It's just a different way of showing the map. Now here's where it kind of gets interesting. Now if we break these down, each one of these dots or triangles is one of those watersheds from the previous map. Okay? So what this says is when the disease is bad and when the nitrogen levels are bad, the footprint statistic indicates they're bad. These are also areas, coincidentally, where macroalgal blooms, nuisance algal blooms, have been chronic problems, and spatially chronic. In other words, they've occurred over a kilometer repeatedly for the last 50 years. We've had problems with this. And then, interestingly, where the disease hasn't been a big issue and the nitrogen levels are low, there haven't been those problems there. So we have this positive correlation with the algal blooms, and we have this negative correlation, both of them sort of indicating that maybe macroalgae plays a role. Well, if you know, green sea turtles eat what? Limu, right? They eat algae. And they actually eat, what they ate 50 years ago is not really what they eat today. Green sea turtles are the only sea turtle species that has, through its whole life cycle, that when it recruits to near shore, they're basically herbivores their whole life. Other sea turtles will eat crustaceans, hawksbills eat sponges, and fireworms, and just a bunch of different stuff. Loggerhead turtles on the East Coast eat horseshoe crabs. If you've ever seen those kind of really ancient, sort of Jurassic-looking things, they are ancient and Jurassic. They don't just look that way. Loggerheads eat those, but green sea turtles are pretty much eating algae their whole life. These are two turtles over just up the road here. Two turtles, 1, 2, and they both have tumors on their eyes. It almost looks like someone took this in Photoshop and shrunk that one and then put it back up there. It's very similar looking. And they're eating this hypnea right here, Hypnea muciformis. Sometimes it's called hookweed, that bright purplish reddish, can get really dark, really rich in nutrients, and they love this stuff. I was just yesterday at Ho'okipa, we were out in Paia in the water, the rocks, and there's hypnea all over, and there's this turtle just going for that stuff. I've seen that up at Honolulu. If the turtle has a choice, and you would set up this metaphorical experiment, you put like cheeseburger and broccoli and hypnea and seagrass, they would go for the hypnea every time. They just love this stuff. It looks like Pteropodiella, if you know that, but that's native. So why would macroalgae, all this stuff, okay, we have land use, we have this disease, we have nutrients, we have algae. Well, what's the link? This is a herpes virus, right? So you have to think like a herpes virus, if that's possible. So I spent a lot of time thinking, how does herpes work? And I read a lot of papers on herpes, I read books on herpes, and I tried to immerse myself in that herpes biology for a few months. And something interesting, before we get to that, is that kind of a truism across plant physiology, from pine trees to corn to tulips to macroalgae, as we found out, is that I mentioned the waters here are oligotrophic, which means there's few nutrients. Well, plants love nutrients, kind of like we like calories, right? And when we take carbohydrates in our body, do we turn those carbohydrates into hair and teeth and toenails and bones? No. When we have it left over, we turn it into fat, right? And plants, when they have nitrogen, is what is really limiting for them. If you give them nitrogen, if you give them fertilizer, they grow, right? That's why we had the green revolution of the 60s and 70s. We start fertilizing everything, we're like, oh my gosh, it grows. Well, when plants have the opportunity, when the nitrogen levels are elevated in the environment, they do not indiscriminately take that nitrogen and just pack it away in all sorts of different places. They package it preferentially away in this thing called arginine. And it's an amino acid. It's one of the 22 amino acids. It's the only one that has 4 nitrogen groups. So it can pack away more nitrogen than the other ones simply because of its molecular formula. Now, plants, they tend to preferentially store, I call this luxury consumption, or nitrogen sequestration is another way to say it maybe. They pack it away as arginine. Well, why is arginine interesting? Well, here's the herpes immersion I was telling you about before. This is a herpes electro micrograph image of a herpes virus, and you might be more familiar with the virus kind of looking like a lunar lander, kind of looks like Apollo 11 maybe. That's HIV. It has that kind of crystalline nuclear envelope, and then a stem, and then these kind of crazy legs. This is a little different looking. Obviously, it doesn't look like Apollo 11. But we have, if you know what a virus does, a virus is kind of like a parasite. It can't make its living by itself, right? It has to hijack its host cells. And what herpes does is, here is its nuclear material, like its DNA. A virus can have RNA or DNA, and what it does is it needs to, in its host cell, it needs to attach, it needs to find the host cell nucleus, it needs to attach to it, it needs to tunnel through that and break that open and insert its DNA or RNA into that nucleus and trick the cell into thinking that it's herpes, and trick the cell into producing more herpes DNA. But in order to do that, it relies on, you can see these little faint little lines right here. These are chaperone proteins that make up this little envelope around the nuclear material. And there's a particularly really important one called glycoprotein L. And this is the one that is integral, absolutely crucial for herpes to be able to find, attach, and enter into, basically do herpes nefarious deed, right? To take over that, to hijack the cell, and to turn it into a herpes factory. But this protein right here is 60% arginine. And arginine is not created by the body. We do not synthesize it predominantly. We can, but we get it from our diet most of the time. So if you don't have arginine, herpes does not basically have the fuel that it needs. It doesn't have the chaperone protein, and the whole factory shuts down. So what they've done in rabbits and in monkeys, they've done experiments on them, and there's this one experiment where they had rabbits with corneal tumors from herpes, and they will get eyedroppers and they will drop this enzyme in those tumors in their eyes, like little eye drops, that basically breaks down arginine. And when they do that, these tumors shrink. So there's been a lot of studies, that's just one, they've been done in a lot of different kinds of studies. They've shown that when you restrict arginine, you can really atrophy a herpes infection. But it's been shown to be very integral for herpes. Now I just mentioned that plants store this away, and so what we think is happening is that the turtles are basically eating themselves sick. They get herpes from bumping into one another, from a fish coming over, just snacked on your buddy's tumor, and now it's snacking on your eye, or just kind of bumping into you. It gives you, we're not talking about this disease. Again, I said herpes is ubiquitous. It's everywhere. Everybody has it. But it's not always promoted. What we think is happening is in certain places in Hawaii that have issues with nutrient management, perhaps from agriculture, perhaps from wastewater treatment, perhaps from natural things like fish ponds, or from estuaries that have freshwater content and a lot of nutrients. What they do is the algae there are elevated in nitrogen, and not just any kind of nitrogen, but they store it away in this arginine. The turtles are eating this, and they are they're basically eating themselves sick. So they're packing away all of this fuel for herpes. So let me just run through some things. We know size is a risk factor for the disease. We know it varies locally through time. We know that this spatial map has these clusters, and it looks eerily similar to this nitrogen footprint statistic we came up with. We think macroalgae kind of has, in history, it kind of has a role. And then there's this theoretical relationship with arginine regulation. So what we've done since this paper I put out, is we've looked at algae here collected in Maui, and we compared Kihei to Olowalu, which is according to my nitrogen footprint statistic, Kihei is a high nitrogen area, and Olowalu is a low nitrogen area. And so what we have here is 3 different algae, and this is a delta N15, and this is a certain kind of nitrogen. This is a certain kind of nitrogen that happens to come through wastewater, through sewage. And what this says is in Kihei, for this algae, the N15 level is really high, which means if you're above about right here, it says it's an impaired watershed. So this is very high. This is telling us that the nitrogen in these algae in Kihei is coming from sewage. In Olowalu, that's not the case, but we wouldn't expect it to be. In Acanthoptera, this is another turtle food, same thing, low level, high level. And this is another turtle food, this is like the sea lettuce, that bright green stuff you see. Again, high in 15. What's interesting is that this one, ova, if you've ever seen it, will actually grow on rocks in the spray zone, almost above the high tide line, right around that. And so it's a little bit different than the other ones, but it's even getting some N15. But that perhaps is from surface stuff, maybe a little different. But what we have this pattern here is that these turtle foods, which we theorized in this study we just did, are actually getting their nitrogen from the wastewater. But are they storing it in arginine, as we predicted they might? The answer is yes. Here is Olowalu on the bottom, on all of these, and the darker line is Kihei, and this is arginine here. These are all the amino acids we tested for. And sort of the way I like to think about this is a rising tide raises all ships. but not equally. Arginine, if it's a ship in that metaphor, it gets bumped up higher than the rest of them. For all of these, there's a spike in arginine. So you can see that not only is the nitrogen coming from wastewater, at least here in these two places in Maalaea Bay, but it's also being packed away and sequestered in arginine, which, if you remember, is herpes fuel. So that's really important. Just today, we went out in Kahikili Beach, and if you know, there are some, you can go out there and see this yourself. The water right here is kind of fuzzy. You don't have to like get the stuff off your contact or whatever, and on your eyes. This is the picture, but it's in focus. It's not a bad picture. It's actually a very good picture. So you can see this area is nicely in focus here. This area is kind of really blurry. I put my hand into this today. I've touched the wounds in the side, right, okay? And this is actually a seep. This is a groundwater seep in Kahikili. And they put a tracer, they put a green fluorescent dye in the sewage treatment plant right here in Lahaina. And they did that last August. And that green dye is now coming out of this reef. So what it says is that sewage is percolating through the groundwater, it was injected in there, through the porous rock we have here in Hawaii, and it's coming out the reef. Here's another picture. I said this kind of reminds me of the smoke monster in Lost, if you remember that. It's kind of like the blob coming right at you. There's the hole where it's coming through, and this is kind of the area right there. There's the big nasty seep. I put my hand in there. It's hot. It's kind of like, it looks like you had hot water and you squirted a bunch of maple syrup in there. It's kind of swirling around. It's kind of viscous. And this picture was taken. This picture is not from today. This is from a colleague of ours, Meg Daler. She took this picture. I think she took this a couple weeks ago, but the dye hadn't begun to come out there. But today, this looked like someone was squeezing out like a gelatinous green Gatorade out the holes today. And this has been established, and we know that the water from the wastewater treatment facilities that's injected to the ground, these sewage injectors, is coming out. It's coming out these seeps in the reef. It seems to be that the macroalgae is sucking it up, the turtles are eating it, and it's giving them this disease. So a lot of people have helped out with this, including Donna Brown over here, and who runs the stranding program for us here on Maui. But I want to show you one more thing before I go, and before I answer some questions, and that's this piece of paper which you might have picked up when you came in. This is a project we're doing that you can help out with. You have photos of turtles that have tumors, or if you have photos of hawksbills, which are a little bit less common, you can upload them to this website. It's a project we're doing with iNaturalist.org. There's even an app for your iPhone or your Android phone. I'm an Android driver, and we have an app on here. I just uploaded one the other day. You can go on there, you sign up for an account, you log in, you upload your observations of whatever. Here you get really good eyes. This is hawksbill sea turtle, hawksbill sea turtle, hawksbill sea turtle, hawksbill sea turtle. And what you can do is just add it to our project. And what we're going to do with this is, when we eat the strandings program, this study I did, what you just talked about, those are dead turtles, right? We want data from turtles that are alive. What about, is that different? There's a lot of different things we want to get into with this, but right now we need your help. There's a lot of people that have good photographs, and one of the things that has been a concern is people say, well, I don't want people to know where the turtles are, because then they might go there and they might harass them and might even kill them. What we can do is, what we have done, is that when you put it in there, it will randomize the location within a given halo of like 10 kilometers. So if you put it in there within a 10-kilometer radius, it'll put a circle in there, and no one will be able to see. So sometimes you go on there, it looks like it's on land, the turtle isn't actually there. I can see where they are because I'm the curator of the project, but the general public can't go in there and see where the hawksbills are. If you go to say, oh, there's some in Makena, but I don't want someone to go there and kill the turtle for a shell. Yeah, we've thought about that already, thankfully. And here's the project. This is a photo of a basking turtle up in the Northwest. Irene, actually, the woman who spoke before me, actually took that picture. And there's a bunch of different, you can see Maui's pretty popular right now. There's been people uploading a lot of photos. Don't have much from Kauai, so if anyone has pictures from Kauai, that'd be awesome. We have nothing from me, of course. But we'd love to have your input, adding photos. You don't have to be an expert. If you upload a photo, you're like, is this a green, is this a hawksbill? That's what it's about. It's basically crowdsourcing this problem for anybody who has photos, but you have to have a photo. That's the thing. Because we can't just trust you. We need a photo to document it. So thank you very much. So here's, I'll just show you some images real quick, and I'll take some questions. This is a hawksbill. Guy from Big Island sent it in. This is a turtle basking over near Pahoa on the Big Island is a turtle up in French frigate, actually tumors on it. We're particularly interested in green turtles with tumors and hospitals. Any questions? Yes, ma'am.
How far back for photos?
However you want. We'll take all comers. You have like old slides, Velvia films. I have those slides. But it really, I mean, don't say, well, I have these pictures, but they're not good enough, they're not high quality. These are some good photos, I'm poaching good photos. So maybe we'd like to know, but we'd love to see your photos on there. Any questions? Yes?
Do you know why the algae became more arginine or the other?
So that's a good question. Why is algae, what do plants in general, or algae in particular, contain, why do they have more arginine? Why does a nutrient-rich environment, why do they pack away as arginine? There's a really complicated biochemistry answer for that, and it just has to do with the feedback of how the plants process nitrogen. When this feedback mechanism gets shut down and just seems to all accumulate in nitrogen. I can give you a really interesting paper to read, but it just has to, there's not an obvious explanation. There's a very technical biochemistry explanation for it. It's just this feedback mechanism gets shut down and it all starts to pile up. Happens to be beneficial, but it makes sense. I mean, it's the only one that has 4 nitrogen in it. It seems you could pack more nitrogen in there than you can in phenylalanine or guanine or something, but it just seems that it's actually just this feedback loop gets shut down.
By like chemical compositions and
You know, a lot of these things have to do with geometry. And these are 3-dimensional chemicals, and they kind of fit together like Tetris in a really complicated way. And this feedback mechanism, I think it probably is not the same across all kinds of plants. But there's a paper by a research group from Barcelona that put this together. It's just this feedback loop gets shut down. It just starts to all back up like logs in a stream. Yeah. Other questions or comments or insights? Yes, ma'am.
Any idea how long once tumors are detected on a turtle, how long it would take it for it to end up killing?
To die?
Yeah.
You know, sometimes it takes a few years, sometimes it will happen very quickly. It'll be a matter of months. I'd say it probably depends on where they are. And often the time these turtles show up, if they have tumors, there's 10 things that are seriously wrong with the turtle. We don't know in what succession those happened. Maybe it became immunocompromised and it had herpes and then all of a sudden it just blew up. We don't tend to see turtles like that are missing flippers from shark bites with tumors. It's not like they get sick and then the shark preys on them. I think the sharks are pretty good at finding them independent. But they have all sorts of problems. They're basically their kidneys start to shut down after a while, and then you probably can imagine that's no good. They often die from pneumonia.
Okay. Do the females, I mean, is there any compromise in them laying eggs if they happen to have the tumors?
You know, I don't think there hasn't been many examples of turtles with tumors that have successfully bred.
Okay.
It does seem to be, it's females breeding and putting all that metabolic energy into yolks is the most costly thing a sea turtle can do. It seems that what we think is that this disease is costly enough energetically that it's not going to do it.
Are you seeing that there's like selection of, they're getting all these tumors before they can breed or before they can breed successfully for a long time and they're returning to the same areas to do this? I mean, I guess there's lots of plants in these areas, lots of nitrogen, so it seems like a good area.
It's like a risky bet, right? Like it seems, yeah. It's like there's all this food here, but you can't eat it.
Yes.
Like water everywhere, but not a drop to drink. Yeah, I mean, it's interesting, you know, not every turtle in the water has tumors. You can go to one of these bads, we analyze strandings. So these are turtles that died. But what about the whole standing population in this spot? What's the real sort of apparent prevalence of the disease? That's a little bit tougher for us to get to because we don't know everything about every turtle. But it is interesting that, you know, there's a lot of food there, but maybe not being beneficial. Yeah, but there's still a lot more work for us to do. For example, we're doing some experiments with the algae. Actually, I told you about some wild collections we did here in Maui. We're doing that on Oahu. as well. But we were also growing algae experimentally in tanks and trying to manipulate them and trying to really spike the arginine levels and see how exactly this happens. Does it work the same for algae as it does for corn, something like that? We're trying to figure that stuff out.
A ballpark it up on the prevalence? I mean,
Today,
yeah.
at one point we think up to 2/3 of the population was afflicted, but that was in the 90s. It was feared in the 90s and I think in the late 80s that this was going to drive this population to extinction and also the Florida population. I will say that this disease happens in, in Caribbean, in Florida, in Indonesia. It does not happen in American Samoa. It does not happen in Guam. It does not happen in the remote islands like Jarvis, Howland, Baker, Palmyra Atoll. Have you heard of that? Everywhere this happens, they have A, problems with wastewater management, and B, problems with invasive macroalgae. We know that for a fact. It's true. These species of algae these sort of problematic species, these species are everywhere in the world. We just recently reclassified the sea lettuce as ulva. It's not ulva reticula, it's ulva lactuca, which is a globally problematic algae. People have thought, oh, that sea lettuce is native Hawaiian. It's not. It's a weed. It's not native. So they have these problems everywhere. Thanks a lot for having me tonight.
You're invited to